Okay, today we're going to continue with the cell cycle, and we're going to actually start with a quick review from last class over some key terms and key concepts regarding just what is the cell cycle meant for. And then we're going to get into all of interphase. We're not going to get into mitosis, I don't believe, but we should get into that tomorrow. All right, so let's get the show on the road. Cell cycle, and more specifically, we're really just going to knock out interphase today, okay? So a quick review. When you're going through the cell cycle, and you're actually going to go through mitosis, do you recall what are the three main jobs that mitosis attempts to accomplish? What does it try to do regarding cells in your body? Anybody chime in anytime? Ryan? Well, for what purpose? Why are you creating cells? What's the purpose? That's what I'm looking for. Okay, let's do repair. Okay, so Ryan gets really super excited because he's 18 years old and he's going to vote for the first time in his life because that is his civic duty. Right, Ryan? That's you should. Okay, next election, right? There you go. Attaboy. All right, so he gets so excited he's going to fall down the stairs and break his arm. Ouch. His arm is going to need to repair itself. Now, he might have to have surgery or might just have to have an orthopedist reset his bone so it can heal properly. But once everything is all lined up and good to go, his natural healing processes will take over, and he's going to have brand new bone cell grow and repair that damage. This happens in our bones, our soft tissues, your skin. So you mainly have repair. What else is there? Growth. Most of you are juniors and seniors, so I think it's fair to say that you have gotten taller and bigger in high school compared to what you were when you entered high school. You have literally gotten bigger. Duncan at 6'2", 6'1", is bigger than he was when he was a 7th grader. He is literally taking up more volume. That means you need more cells. And what is the third one? Replacement. Even when you guys get to that point in your lives where you stop growing, you still have to replace cells because cells have a lifespan. And their lifespan is not as long as your lifespan. Skin cells last a month. They go from the bottom of your epidermis and it takes a month to get it to the top and they fall off. They shed off. Some cells last longer than others, but more or less they just have to be replaced. Fun fact, every 3 seconds, about 40,000 of your cells are just wiped out. But remember, they are replaced. All right, so that's mitosis. Do you quickly remember what meiosis makes? Jasmine? Okay, it is the genesis of reproductive cells. Your sperm and your egg. Remember that females are born with all the egg cells that they're ever going to have. Ladies, you're born with several thousand, but by the time you get to puberty and you start your period, you're probably down to just a few hundred. And you will eventually run out. Guys, you do not begin producing sex cells until probably 12 or 13 years old, and you will not stop until you die or something happens to your ability to produce sperm cells. All right, now, as far as key terms are concerned, we went over 4 of them. First one is somatic cells. What type of cells are somatic cells? Okay.
Body cells.
Body cells. Any cell that is not a sperm and any cell that is not an egg is considered a somatic cell. A stomach cell, somatic. Liver cell, somatic. Brain cell, somatic. Arm cell, bone cell, cartilage, tendon, ligament, kidney, uterus, all of them, somatic cells. What are gametes? Sam? Sex cells. Now, you guys will need to get familiar with the gender symbols. It's going to make it a little easier for this class. Here's the female symbol. The female sex cell are eggs. And here's the male symbol. That is going to be sperm. Fun fact, the female egg cell is the largest cell of the human body. You could actually see it without a microscope, but you got to be up real close. The sperm cell is the smallest cell of the human body. Okay. Next we have diploid. Its symbol is 2N. This is going to be 2 chromosomes per pair. For humans, what is our diploid number? Our diploid number of chromosomes. Jasmine? No, that's not our diploid number. No, it's not 24. Anybody? Not 23. She said 23, not 23. Duncan? 46 is our diploid number. N is the minimum amount of chromosomes that you need to survive. 2N is double that. Our diploid number for humans is 46. We have 46 chromosomes. We inherit 23 from mother. We inherit 23 from father. They pair up within our cells, and that makes 46 even. Ryan, what's your favorite animal or plants or fungus or anything? Sure. Shirataki mushroom. Let's see if I can find that out. How many chromosomes in a shirataki mushroom? It's you sure it's shirataki, not shiitake?
It's my favorite. Shirataki now.
It is shiitake. Looks like there's only one set of chromosomes. I'm trying to get more. Does that mean there's one or is there two? How many chromosomes are there?
I thought I knew, but apparently that's not.
Looks like there's 8. So your shiitake mushroom is 8. Let's see. Hannah, what's your favorite plant? How do you say it? Racco? Are you saying Rapto or Graco? What's the second word? Petulum? I totally did not spell that right. Petulum. I got the second word. I still couldn't hear you. You're saying, what's the first word? Grapto. Oh boy. All right, I found it. Let's see. Looks like it's about 26. Dan, take it easy on me with this. What's your favorite animal?
Bengal tiger.
A Bengal tiger. Good call. Somebody say bird. Yeah, because that's all. As far as I know, there's only like 5 species ever: shark, bird, tree, fern, ant. Bengal tiger has 38 chromosomes. No, because there's hundreds of thousands of species of bird.
What about like a pelican?
I'm gonna start calling you Bambi. Bird. All right. Haploid is one chromosome per pair. Don't worry, Ryan, when I post this on YouTube, you can watch this and listen to yourself. All right. For humans, our haploid number is what? 23. What is the haploid of a shiitake mushroom? 4. How about a graptopetulum? And how about a Bengal tiger? Oh, mental math, oh boy.
19.
So remember that as humans, we have 23 pairs, but the definition of a pair is 2. So 23 times 2 is 46. You're going to see things like this on your exams and on your tests. It's going to say, you have a platypus. A platypus has 30 chromosomes. What is this haploid number? 15. Haploid is always half the diploid. Okay. All right, these are the 4 terms we did yesterday, but now let's get into it. You guys are going to get a whole new set of terms you'll need to study over the next 24 hours. I want to remind you that this unit is like a bridge between cells and genetics, and there is a ton of vocab in genetics. So you need to stay on top of your vocab on a daily basis and not let it bury you, because there is a lot. All right, let's begin. Cell cycle. Interphase. We got into this a little bit yesterday, but we're going to get all through it today. Majority of the cell cycle is spent in interphase. In mammals, interphase can represent up to 90% of the entire cell cycle. When we say inter, it means in between. Intermission in a lightning game, intermission during Gone with the Wind, because it was such a long movie, intermission during a dance recital. So this is in between cell division events. The cell just got finished dividing, and it might just go back into another round of division. Interphase is composed of 3 stages: G1, S, and G2. We're going to talk about each of these individually. Any questions so far? All right. Okay, let's do G1 phase. G stands for growth. There's a lot of things that are growing in this phase. The cell is going to nearly double in size. Why is that, Mr. Thorson? Good question, Dan. The reason why is because the cell is going to double its number of organelles. Why is that, Mr. Thorson? Dan, you are full of questions. The reason why is because the new cells that you're going to make need to look identical and be identical to the original cell. If the original cell had 12 mitochondria, you need to be sure that each of the new daughter cells is going to have 12 mitochondria. So you're going to have to double what you had. Okay. So other than replication, or excuse me, other than doubling the organelles, the cell continues its normal behavior. And that would include things like cell respiration, and communication, just to name a few. Okay, I'm going to show you guys a visual as to why it's important that you double the organelles. Some of you may get it. Some of you may do better with a little visual. Happy to do that based on past years. Sometimes this is hard to understand. So let's see, the pen's moving. Okay, so check this out. Let's say that we have a parent cell right here. This parent cell is the cell that you want to copy. And within this parent cell, I'm going to tell you there's 20 mitochondria. Let's say that there's 5 ER, endoplasmic reticulum. There's 12 Golgis. And let's say that there's 1 million ribosomes. Well, the first thing that the cell is going to do when it goes through G1, it is going to get bigger. Nearly double in size because it is going to double its organelles. So now it has 40 mitochondria. It has 10 ER. It has 24 Golgi. And it has 2 million ribosomes. The reason it does this is when it goes through mitosis, which we will likely not get to today. We'll get to it tomorrow. And it actually divides into 2 daughter cells. The 2 daughter cells are going to have 20 mitochondria each. 5 ER each. 12 Golgi each. And 1 million ribosomes each. These are the daughter cells. The daughter cells are expected to be identical to the parent cell in nearly every way. If the parent cell is diploid, the daughter cells are diploid. If the parent cells are a bone cell in your humerus bone, then the daughter cells are bone cells in the humerus bone. But this is why you're going to double everything. Are there any questions with G1? Yes, Sage. I can't understand. Speak a little louder. Because this is going to be very temporary. As far as the conservation of the space and conservation of matter, there are signals that are going to be sent to the nucleus to generate the proteins from the DNA that are responsible for making these organelles in the first place. And it's just going to be very temporary while this, or for this cell to have that many. The cell's not going to be doing a whole lot of metabolism. It's currently in just a state of division. It's not going to be doing a whole lot of other activity once it has up to that number. Now, in G1, it's important for me to include what G0 is, or G0. This is going to be an arrested state. Right now, all over the city of Tampa, Florida, there are fire stations, and there are firefighters and emergency medical personnel that are working in those fire stations. And some firefighters have 12-hour shifts, some have 24, 36, maybe even 48-hour shifts. And if there's not an emergency going on around town, perhaps they're doing drills to make sure that they're ready when the alarm goes off. They might be watching ESPN, just kind of relaxing. Maybe right about now they're having some lunch, or they're just making sure the fire truck is gassed up, ready to go. All their equipment is in proper order, ready to go. But more or less, they're not doing much outside the firehouse. And then when the signal goes off that they need to go jump into action, they spring into action, jump in the fire truck or the ambulance, and off they go to save the day. Not every firefighter is putting out a fire 24/7. You only have to do it whenever it's called for, whenever the call comes in, whenever there's a signal. Well, not every cell is going to be going into the cell cycle and dividing over and over and over. Sometimes you're just going to chill. And so cells that are not currently dividing or in the process of division are said to be in the G0 state. However, a signal must be sent and received in order to bring the cell out of G0 phase. And there also must be signals to put it into G0. But a cell that is not actively dividing or in the process of the cell cycle is said to be in the G0 or G0 state. Okay. Next, let's do the S phase. S stands for synthesis. The DNA is double. Why would the DNA double? Duncan, can you tell me? So that the daughter cells will receive the same amount of genetic information that they're supposed to be in the parent cell. So this is where you guys are going to get a few more terms here, all right? We all know what DNA looks like. It looks like this when you're really zoomed in on it. Well, if I were to zoom out, it would look like this. This is called chromatin. Chromatin is decondensed DNA. Think of it like spaghetti on a plate. It's all spread out all over the place. Well, when you're going through mitosis, it will condense, and it's going to look like this. This is called a chromosome. As humans, we have 46 of these little boogers. This is condensed DNA. This is like taking your fork into a plate of spaghetti and twisting your fork, and so the spaghetti wraps around your fork to condense. I want you to look closely. See how that little line, all the lines I drew? That's DNA. It's just highly, highly condensed. It's tightly packed. You know, some girls have really, 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 really long hair, and they tie it up to a little bun. All that really, really long hair is tightly packed into a little bun on top of their head. That's kind of like how a chromosome is. All the hair down to their butt, that's chromatin. When it's all tied up in a little bun, that's chromosome. Yes, Ayla.
So are you saying that
The chromosome is composed of condensed chromatin, and the chromatin is composed of a single DNA strand. The whole thing is one chromosome. Yep, that's one whole chromosome. Okay, so here's what happens during the S phase. You ready? I think it's the effect of an E. All right, so we have chromatin, okay? Just a huge pile of DNA. Just all that is just one DNA strand and just a huge hairball of chromatin. But then we're going to go through the S phase. Oops. Perfect. Okay. Now, this is really important to understand, guys. You have two copies of the same DNA. It's identical. Two copies of the same DNA. I know it's tough for you to do that on your notes. Try to make them look as similar as possible. You once said, perhaps this is going to become chromosome 1. Well, now you have two copies of chromosome 1. All right, well then when you go through mitosis, and in mitosis, it's going to condense. Now you have two, whoops, come on. Why is it doing that? You have two chromosomes of the same type. In other words, you have two copies of the same chromosomes. You have two copies of the same chromosome. Now, you don't want to have two copies of number 1 of the same type. You all have chromosome 1 from your mom. You have chromosome number 1 from your dad. That's how it's supposed to be. Chromosome 1 from mom, 1 from dad. 2 from mom, 2 from dad. 3 from mom, 3 from dad. It goes all the way up to 23 from mom, 23 from dad. All in all, it's 46. Well, you don't want chromosome 1 from dad, chromosome 1 from dad. That's a mistake. And so in your cells, you need to make sure that these divide. So what actually happens within your cells during mitosis is that these two will be bonded together in a way. These are called sister chromatids. Sister chromatids are two copies of the same chromosome connected. And you're going to later rip them apart. We're going to get into that tomorrow. But you see this little red structure and this orange structure I drew? I'm going to identify and define those for you so you know what they are. You're going to need to know that for tomorrow, so might as well just tell it to you now. The red structure in the middle is called the centromere. You see the word centro in there, like Centro Ybor. You want to go to Tampa Bay Brewing Company for dinner, have a little birthday party down in Ybor City for your mom. You go to Centro Ybor, the center of Ybor City. This is the center of where two chromatids meet. And then you have these two little orange structures here and here. These are called kinetochores. This is where spindle fibers attach. Spindle fibers are kind of like a fishing line, and they're going to rip these two apart from each other so that each new daughter cell gets one copy of this chromosome number 1. When we do mitosis tomorrow, you're going to see a bunch of sister chromatids. We're going to divide it into 2 daughter cells. That's going to be for the next lesson. But this is the purpose of why we do the S phase, so that you have 2 copies of the same chromosome in DNA, so that the daughter cells will get as much DNA as they're supposed to get. So if you think about this, here's a parent cell, and let's say this is Duncan's skin cell. He's going to give himself a nasty little paper cut today on accident. He needs to regrow some skin cells. Well, his parent cell, the skin that's still there, has 46 chromosomes. It's going to go through the S phase. And so for a very, very, very brief moment of time, after his cell goes through the S phase and doubles the DNA, how many chromosomes is he going to have in that parent cell? 92, just for a minute. You then go through mitosis, and you're going to have 46 and 46. These are your daughter cells. But this is why we do the S phase. Yes, Ayla? No. First one, then the other. No. What's they?
So the cell has double the organelles.
Yes.
Now doubling its DNA, and
After it doubles the DNA, it goes through G2, and then it goes into mitosis. Yeah, Annie? What? Spindle fibers attaches. The spindle fiber is like a fishing line that grab onto both sides of the chromatid and pull them apart. You okay, Ryan? I'm staring off into space there. All right, let's get into G2. G2 is pretty lame. There's not a whole lot going on here. In G2, you're going to synthesize proteins that aid in cell division. And you're going to make the final preps for cell division. Cell division is a very nasty process. It's not very graceful. It's pretty brutal. You want to make sure the cell is prepared for it. Splitting into 2 is not a fun thing. It's actually a very violent act. All right. And very briefly, we will mention the mitotic phase. This is the M phase, or the mitotic phase. This is not interphase. Interphase is over by the time you get to M phase. The M phase includes nuclear division. The nucleus divides. And it includes cell division. Nuclear division is mitosis. Cell division is cytokinesis. These include prophase, and we'll do these tomorrow. Metaphase, anaphase, telophase. That's going to be nuclear division. And then cell division is going to be cytokinesis. Okay. Are there any questions about any of the stages of the interphase and the M phase and the goings on that happens in each of them? We will delve into mitosis, which is the M phase, tomorrow. We're not done yet. Are we done? No, we're not done. We got 8 minutes. So if you were to actually look at the cell cycle, it would appear like so. All this is going to be interphase. Yeah, G1, S, G2. Oops. The orange region is the M phase, and in the M phase is mostly mitosis. You have prophase, metaphase, anaphase, and telophase. That's going to be mitosis, and then at the very end you have a C I'll put for cytokinesis, where the cell actually divides in two. Prophase, metaphase, anaphase, telophase, cytokinesis. This is all mitosis. Cytokinesis technically is not part of mitosis, but is the last part of the M phase. Mitosis includes the pro, the meta, the ana, and the telo. Okay. Now, let's move on to cell signals. This is going to be pretty important. The cell cycle needs to be regulated. It needs to be controlled, and it needs to be instructed. Signals are going to be responsible for that. The cell cycle is regulated by signals. Signals instruct the cell when to proceed with the cell cycle, move on from stage to stage, or to stop the cell cycle when something is wrong. We're going to go over what those wrong things would be. And when it comes to stop, we have 3 checkpoints. These checkpoints are to check up on the DNA. And the 3 checkpoints are located at the end of G1, at the end of G2, and at the middle of M phase. Now let's see if you guys may be able to pinpoint, of all places of the cell cycle, why would the checkpoints be in these places? So I'm going to put them up here on the cycle. The first one is right here. That's going to be checkpoint number 1. The second is going to be at the end of G2, checkpoint number 2. The third one is going to be at the end of metaphase in mitosis. That is going to be checkpoint number 3. So end of G1, end of G2, at the end of metaphase, which is pretty much the middle of mitosis. So do any of you have any idea, of all places, why would you have a checkpoint at the end of G1? What are you trying to check up on? There it is. Start. You make sure you have the right number of organelles. That's a good guess. It's not what you're looking for. Dan?
Make sure it stops growing.
Make sure the cell stops growing so it doesn't get too big because you're doubling the size. That's a fair thought. That's a good thought, but not it. What came before G1? What was the cell doing before G1? Remember, this is a cycle. What was it doing before G1? It was what? No, not G naught. It was splitting. Before G1, you did metaphase. Look up here. Look at what's before G1. Metaphase. It split. Splitting is a nasty business for a cell. And so you want to be sure that the DNA survives the division process. You want to make sure the DNA is okay before you go forward. You know, in factories, they have checkpoints. Tire factories, toy factories, televisions, computers. They have a checkpoint to make sure that you're building something correctly before it moves on to the next stage. You don't want to build a big, beautiful computer only to find out that it should not, when there's like 50 steps to build an Apple computer, and you find out that after step 2, there was an error, but the error didn't get caught. And so now you have a big computer with an error built in it that should have been caught 48 steps ago. That's a problem. You want to be sure that the DNA is okay to move on to the next level. And if not, you need to do something about it, whether it be fix the problem or destroy the cell entirely. The next one is at the end of G2. Before the G2 stage is the S stage. And in the S stage, you double the DNA. So you want to check DNA to be sure it replicated. And it replicated appropriately. Was there any damage in the DNA after it replicated? You're going to find out. And then something that you're going to find out tomorrow. You may not remember what happens in metaphase or after metaphase, but you want to be sure that the DNA will be evenly divided. Yes, Ayla.
So, like, genetic mutations occur when the checkpoints are working?
Yep, you stole my words. That's exactly what I was going to get to. We're going to talk about mutations tomorrow, but the bell's going to ring any second, so I'm just going to try to get this out. Life is dependent on mutations, and the evolution of life is dependent on mutations. You can't stop it. These checkpoints are trying to stop mutations, but they are not 100% foolproof. So that's the last entry I'm going to put in here. These checkpoints are not 100% foolproof. If they were, we would all still be bacteria. But we're not. So I think this is a good place to stop today. We're going to get into this tomorrow. We're going to begin tomorrow talking about mutation and cancer a little bit.